Hello, welcome to another screencast where this time we'll be discussing viruses and their kind of quote-unquote life cycle and how they can contribute to, a little bit about how they can contribute to some genetic variation within their host cells. So by the end of this you should be able to describe the basic structure of a virus, the basic viral reproductive cycle. We'll compare the lytic and lysogenic cycles and the cycle of an enveloped retrovirus, HIV, which I'm sure most of you have heard of as an example of those. And also then be able to explain how viral replication differs from other reproductive strategies in terms of asexual or sexual reproduction, how viruses introduce genetic variation into the host organisms. We'll actually deal with this a little bit more in another screencast and in class. And then how viral replication introduces genetic variation into a viral population. Okay, so let's get started here. So as we see by the title here, cells are small, but viruses are smaller. So if you look over here, most plant and animal cells, sort of on this scale here, and viruses down here to within on the nanometer scale, right? And obviously not visible with a light microscope. Right? So viruses are going to be infecting host cells, and part of their life cycle or their reproductive cycle involves invading a host cell. So here we have a virus, and here's a bacterium, a prokaryote, and by then, for comparison, here would be a eukaryotic cell. Right? And so over here you have an electron micrograph just of viruses, phages infecting a bacterial cell. So this gives you an idea sort of the scale and the size. And so I always keep this image in my head when I'm sort of thinking about these things. So tobacco mosaic virus, or TMV, was the first virus to be discovered. And you see here that it is a long cylindrical virus. And here are several of the TMV viruses that were isolated from tobacco leaves, and this is what happens to these tobacco leaves when they are infected with viruses. As you can imagine, tobacco is a very important cash crop, and so having viruses that are infecting them was causing a great deal of problems. And so the farmers really wanted to know what the heck was going on here with these. So isolating viruses from these tobacco plants was the first step in doing that. Now, viruses are composed of a nucleic acid core surrounded by a protein capsid. Some viruses also have an external membranous coat. So here is TMV over here, and we have TMV as its nucleic acid is an RNA, right? So this is a single-stranded RNA, so there's quite a bit of variation here. In the adenovirus, which causes the common cold, its nucleic acid core is actually double-stranded DNA. Here is this membranous envelope on this protein, so there is a phospholipid bilayer here. Then it has a protein capsid as well, and then this is an RNA virus, and then there's a DNA virus over here, and these phages, the bacteriophages like T4 as the example, have a rather elaborate protein capsid structure there. Okay, so let's take a look at sort of what happens here when a virus infects cells. So viruses are efficient, but they're sloppy intracellular parasites. Right? So here is the protein coat, or the protein capsid, right? And that protein is going to be typically complementary to proteins on the surface of the host cell. Right? Proteins on the surface of the host cell, and so this capsid will in some way interact with receptors on this host cell, and then some of those capsid proteins get taken into the cell, but really you're worried about this viral DNA getting into the cell. Once this viral DNA gets into the cell, the host cell will replicate that DNA using DNA polymerase. right? The host cell DNA polymerase here, and you get several copies of that viral DNA. The DNA is also then expressed, right? So the genes that are on this viral DNA, right, are the genes for more capsid proteins. And so the host cell is going to transcribe to this mRNA and then translate and get more of these capsid proteins. And then the third step here is there's then a self-assembly of the new virus particles, and then they exit from the cell. Right? And so then these can then go to infect other cells, to infect other host cells. Right? Now, this leaving of the host cell by these viruses oftentimes will kill the cell, right? Or at least at a very minimum will limit host cell gene expression, right? Cause altered gene expression, etc. Right? So you can imagine that these cells here dying, right? because of this virus. Right? So there is a animation here, virus life cycle, sort of a little blurb I heard on NPR that I posted there for you. So what we just described was basically also known as the Lytic Cycle. Right? Viral replication produces many progeny simultaneously. So again, take a look at this diagram here and work your way through it. We're showing you here a T4 bacteriophage that's going to infect E. coli. We had seen the Hershey and Chase experiment utilize T4 bacteria T4 phages in E. coli bacteria to determine the DNA was indeed being injected into the cell. So here we see the injection of that DNA. The empty capsid or ghost then is left there. The cell's DNA in this case is hydrolyzed. Right? And so this is a little bit of a variation on a theme, but this viral DNA gets in there. The host cell then will make many copies of not only the DNA, right, but will also make many copies of the tail, and so these are all the capsid, and then there's going to be this self-assembly here, and then you have the rupturing of the cell over here, or the lysing of the cell. Okay. So the lytic cycle is basically what we had described there, but there's also a lysogenic cycle, which is similar but different. So the lysogenic cycle, here we have our phage or our virus docking on the surface of the cell, injecting the viral DNA, but in this case the viral DNA does not lead to the hydrolysis or the breaking down of the host DNA. And so we see what happens here is that the viral DNA gets inserted into the host genome, right? And so this is now a recombinant DNA because it has viral DNA as well as the host DNA. And when the viral DNA is injected, or is spliced into the host genome, we call that now a prophage. So the prophage refers to this DNA segment right here within the host genome. So that's the viral part of that recombinant. And so then what happens is the bacterium is going to reproduce, right? And every time the bacterium reproduces, right, you get many copies of the same thing, right? And this can lay dormant here. So dormant, right, and not really infecting, or not being expressed, not affecting the host cell in any way. But then occasionally, right, some sort of environmental cue signals the prophage to exit, and then it signals, and so if it's happening in one cell, it's happening in all of these cells because these are all clones of one another. So if the environment is signaling this cell, it also signals this one and this one and this one, and you then get this prophage coming out. It's going to be expressed, and you make more viruses, and then the cell is going to lyse and enters that part of that lytic cycle, right? So this is a tricky way that kind of viruses can sort of hide out in the host genome and then reproduce when there is some sort of environmental cue. Right? So lytic versus lysogenic cycle, similar but different, especially the end of that lysogenic cycle there is going to be pretty similar to that lytic cycle at the beginning. So let's talk about these enveloped viruses, and again a variation on a theme, and we'll talk about here HIV. HIV is an enveloped retrovirus infecting CD4 helper T cells in the immune system. Now, this enveloped virus, here's the viral envelope, and this is a phospholipid bilayer, right? And they have these glycoproteins, which are receptors, right? Or actually no, these aren't the receptors. These would be like the ligand. And then on the host cell, you would have receptors that those ligands would bind with. And in the case of HIV, it's going to dock with that, and then the phospholipid bilayer of the virus becomes continuous with the phospholipid bilayer of the host cell. When it does that, and so via this endocytotic method, sort of here you get the capsid entering. The capsid then promptly dissociates, and you're left with now two of the single-stranded RNA molecules in this virus. Now let's go back over here and just take a look at that for a second. So there's two identical strands of RNA, and they're also have this enzyme called reverse transcriptase. And reverse transcriptase is very descriptive of what happens there. So if you had a transcription, you remember, is using DNA as a template to make an RNA. Right? That's transcription. So reverse transcription would be using RNA as a template to make DNA. Right? And so this is what we see happens here. Right? So here's reverse transcriptase. It's going to here do the reverse transcriptase, going from RNA to DNA, and then we use the DNA to make another complementary strand of that. And again, this is all using the host cell's enzymes or the host cell's machinery. Right? So the DNA polymerases are involved here, and then that DNA gets inserted into the nucleus, and then gets inserted into the host genome, and is as a provirus, right? And so this again can lay dormant, and then on some sort of environmental cue. I gotta learn how to write better with this thing. Some environmental cue here. The provirus is then transcribed to RNA. That RNA holds the code, right, to make more of this reverse transcriptase, right? And you make more of the RNA here. You make more of the mRNA also is the code for more of these the capsid proteins here. This stuff all self-assembles and then exits the host cell, and you get a new HIV virus here, ready to go and infect other cells as well. So again, clicking on this animation will help describe that as well. So what makes HIV such a deadly virus is, one, the receptors here, these glycoproteins and this mRNA code mutates pretty highly at a relatively high rate. And so you get glycoprotein variations here, which makes it difficult to fight the virus. And the cells that are mainly infected here are the CD4 helper T cells in the immune system. And we'll just show you this quick diagram of the immune system. And I want to point out just here right now that the helper T cells are right here in the middle. And you notice that these helper T cells, being in the middle of this diagram, right, they're going to stimulate these guys, these cytotoxic T cells, these B cells, which then lead to like antibodies being produced and active cytotoxic T cells, and these are the cells of the immune system that are actually fighting viruses and other bacteria and things. So this helper T cell is sort of a keystone cell of the immune system, and so HIV is wiping out this cell, and you can see now why this leads to then AIDS, or an acquired immunodeficiency syndrome. Immunodeficiency, right? That you're getting a deficient immune system because you're knocking out these cells here, and then you're not stimulating as many cytotoxic T cells, you're not getting as many antibodies, and your immune system basically crashes. So, okay, so now you should take an opportunity here to just review the basic structure of a virus, the capsid and some sort of nucleic acid core, the basic viral reproductive cycle, being able here to talk about the difference, describe and explain the differences between the lytic versus the lysogenic cycles, talk briefly here about HIV, and how this viral replication is different from other reproductive strategies in that it is not an asexual reproduction, it's not a sexual reproduction. You need a host cell in order to make more viruses. How viruses introduce genetic variation, we're going to deal with this in another screencast, and how viral replication introduces genetic variation into the viral population, again mainly mutation here and some sloppy copying. All right. Thanks for listening. I hope that was helpful. Bring your questions to class.